Biology Sec 5. Remember, collect information and prepare slide.
My name is ศิลาชลเพ็งพิษ
2. กิตยานีสัจจาผล Next, province research information.
Hello, my name is Banita Chongkern.
Hi, my name is Hamdani Sameh.
Hello, my name is แสงตอนปูกองชนะ
Link to research of interest, which research matches the content of document 4 and 5, cell structure and function. Okay, today we will present the research that a new technique visualizes mechanical structure of the cell nucleus. Methods could improve in the understanding of fundamental processes of life. Published on January 18, 2024 by the Medical University of Vienna. Alright, we are ready. Let's begin.
An international research team led by Miss Unite Vienna has developed a new technique that for the first time allows detailed observation of the mechanical properties inside the cell nucleus in living cells. This breakthrough makes it possible to track the nucleus' unique dynamic structural change, which are vital for cell function. Published in Nature Photonics, the study offers new insights that could deepen our understanding of the fundamental processes of life.
Researchers note that the mechanical properties of the cell nucleus must be precisely regulated for proper cell function and can change in certain disease. The nucleus can act like both a liquid and a solid, but it remains unclear how this change enables key process decision as nuclear reorganization during cell division or the fast, efficient synchronization of activity across different nuclear regions.
A team led by Karen Alzett at Medical Vienna had developed a method to image complex mechanical properties of the cell nucleus in living cells over time using bright-line light scattering. We analyzed light scattered by thermal vibration to determine elasticity and viscosity. They measured these properties from all angles to create a spatial map of the nucleus. The results show that the nucleus is highly dynamic and has a unique long-range order that may help synchronize its internal process.
This is a video that explains how blue and light scattering from YouTube THA Tech Innovation Channel. If you are ready, let's go see.
Hello and welcome to this video. I'm Thomas from VETEC Innovation, and today, as you can see, I'm in a typical optics research lab. And as you might have already guessed from the title, this is a Brillouin light scattering setup, and in this video I will talk about the experimental realization and our solutions for Brillouin light scattering. So first of all, I would like to mention that this video is about the experimental realization of Brillouin light scattering. If you want to know more about the physical background, the advantages and what it can be used for, you find a very detailed review from some of our company founders in the description below this video. There are also links to some application notes if you want to know about the applications and also how time-resolved brilliant light scattering works. Brilliant light scattering, or in short BLS, is a very powerful tool when it comes to the investigation of phonons or magnetization dynamics such as spin waves. And it was also my main investigation tool when I was still working in research, actually in a lab very similar to this, a bit different configurations, and I was using a BLS microscope, but I will talk about the different configurations later in this video. Since brilliant light scattering works in the frequency domain, detecting frequency shifts of scattered laser light of only a few gigahertz, one of the most important tools is the interferometer. For this, the Tandem-Fabry-Perot interferometer from JRS, which is the black box right over here, is one of the standard tools. I'm not going into the detail on how these interferometers work, but basically it's just two pairs of Fabry-Perot interferometers which are being scanned and need to be constantly stabilized using some reference light. If you want to know more about this, there are also some more details in the review article in the description below. But these devices are quite complex and need a lot of manual alignment. So if you want to work with them, you need a lot of training and experience.
And this application note on time resolved Bill-Wang on the THA Tech website, which can be opened and read, will tell all the details.
The Understanding the Development of Diseases study leader Karim Ilyasad describes the new imaging technology as groundbreaking, revealing unusual nuclear properties that may explain how the nucleus synchronizes vital processes quickly and efficiently, despite its seemingly disordered appearance. The finding could improve understanding of disease-related changes in nuclear processes, and future research will aim to identify the molecular and environmental factors behind this unique mechanical and structural feature.
Okay, next, the summary. The cell nucleus, considered to be the control center of cellular processes, but the material property continue to puzzle scientists. An international research team has now developed a new technique that proves a previously untenable view of the mechanical property inside this control center. For the first time, it has been possible to visualize over time its peculiar dynamic structural feature. A living cell which appear to be crucial for cell function.